Hi, and welcome to our 5th video in the PCDH19 Alliance series of instructional videos on basic genetics. My name is Dr. Kristy Donnelly, and in this video we will be learning about interpreting genetic test results. As I mentioned in our 3rd video on X chromosome inactivation and mosaicism, changes to genes that occur spontaneously are called mutations or variants. You might also remember from our first video that genes are made from a complex structure called DNA, which is an exceptionally long molecule that looks a bit like a twisted ladder. The rungs of the ladder are made up of pairs of letters, A, T, and G, C. The order of the A, C, Gs, and Ts in the DNA is the code that tells the cell and organism how to develop. Many changes in the genetic code of the PCDH19 gene have been found to cause PCDH19 clustering epilepsy. These changes are often referred to as pathogenic variants, as they involve changes to genes that can affect health. A molecular diagnostic laboratory is responsible for sequencing yours or your child's DNA. If a PCDH19 pathogenic or likely pathogenic variant is identified, the variation is recorded on the report from the testing laboratory. Sometimes it is not known if a variant causes disruption to the normal function of the PCDH19 gene or protein, so these changes are referred to as variants of unknown significance, or VUS for short. You may have been given a genetic test result that looks something like the following example. This is a missense variant whereby the DNA code has changed from a G to a C at position 1672 of the PCDH19 gene, resulting in an amino acid change from asparagine to histidine at position or codon 558 of the PCDH19 protein. A codon is a 3-letter sequence of DNA code that corresponds to a specific amino acid. Exon 1 refers to the region of the PCDH19 gene where this change occurred. PCDH19 is made up of 6 exons, with exon 1 being the largest. This part at the end denotes the reference PCDH19 sequence that was used as a comparison for the test results, as it outlines the correct order of A, T, Cs and Gs that make up the PCDH19 gene. The order of A, T, Cs and Gs is also referred to as the nucleotide sequence. As we learnt in video 4 on genetic variants, a missense variant is like a misprint in a recipe. For example, add some rice could be changed to add some mice. The genetic code is changed so it no longer codes for the correct amino acid, and the function of the resulting protein may be affected. You may have been given a genetic test result that looks something like the following example. This is a nonsense variant whereby the DNA code has changed from a C to a T at position 2656 of the PCDH19 gene, resulting in an amino acid change from arginine to a termination signal at codon 886 of the PCDH19 protein. The termination signal can be denoted as an asterisk or by the letters TER. Nonsense variants are like a full stop being introduced into the middle of a sentence making up a recipe. For example, add some rice could be changed to add some. The gene sequence now has a stop signal where it shouldn't be, and amino acids are not added to the protein after that point. This means the resulting protein will be shorter than expected and its function will be affected. You may have been given a genetic test result that looks something like the following example. Here, the DNA code has 8 nucleotides deleted from position 518 to 525 of the PCDH19 gene. This is known as a gene deletion, whereby part of a chromosome or a sequence of DNA is left out during DNA replication. You can think of this like a part of the recipe being left out. For example, add some rice could be changed to add some ice. Duplications are almost the opposite, such that this involves the production of one or more copies of part of a chromosome or a sequence of DNA. Again, linking this to our recipe analogy, we could end up with the instructions add some rice. Deletions and duplications often lead to what is known as frame shift variants, which are changes that mean the recipe cannot be read properly since the letters have moved and the sentences no longer make sense. In this example, the result is an amino acid change from leucine to proline at codon 173 of the PCDH19 protein and a termination signal being added 50 codons after this change. The genetic code is now out of sync and the wrong amino acids are selected to make the protein. This means the resulting protein will not function or may function differently. This is the last of our videos in the series on basic genetics. We hope that you have found them helpful. Check out our next video series on PCDH19 clustering epilepsy to learn about the different outcomes that can be associated with changes to the PCDH19 gene and to continue the PCDH19 Alliance video series. See you then!